Recall from the anatomy of the cochlea, the inner ear, there were different regions or different chambers of that cochlea. It was a spiral organ, right? And what you had was that you had the scala vestibuli, the scala media, and then the scala tympani. Okay, now remember that when we looked at the anatomy of the cochlea, we said that the scala vestibuli, this chamber here, is continuous with the scala tympani, and it meets at this sort of structure here at the end of that spiral organ known as the helicotrema. Okay, so see how these two are connected with one another. And then inside of the scala media, you had that organ of Corti, right? That organ of Corti. So inside of here, the scala vestibuli, the scala tympani, there is a fluid known as perilymph, right? And then inside of here, there is a fluid known as endolymph, right? And so what happens, right, is remember you had your tympanic membrane, and then you had this sort of the malleus connected to that, then you had the incus connected to that, then that was connected to the stapes. So that when this vibrates, and then the stapes vibrates, and the stapes is connected to the cochlea at this structure here known as the oval window. So that is our oval window. Okay. And there's a little thin membrane there, so that when this vibrates, that little membrane vibrates, and it's going to vibrate that perilymph, that fluid, right? And so when we push on this, it's going to push all of that fluid. So if we push in here, draw that in a different color, right? If we push in there, it pushes out here at this structure here known as the round window, which is also covered by a thin little membrane. And so what happens is that this perilymph is going to sit here and it's going to vibrate, and it'll vibrate sort of up and down a little bit. And what will happen is we'll cause this endolymph here to vibrate. Remember, you got that organ of Corti. And that organ of Corti had hair cells on it. And so let's draw one of these little hair cells, like a little candlestick. And I'm going to draw another one next to it. Like so. Because remember, those hair cells had these long cytoplasmic extensions on it, those stereocilia. Now, those stereocilia are connected to one another via like a spring known as a tip link. And that tip link is connected to a channel. So that when this sort of bends, right, what's going to happen is if we bend that stereocilia to where it sort of sends at an angle like this, right? What's going to happen over here is that the tip link, if it's connected to a door, is it's going to pull that door open. And that door serves as a gate for potassium. It's a potassium channel. And so when that gate opens up, because this has tilted, that potassium channel opens up, and we have a super high concentration of potassium inside of that endolymph. That potassium is going to influx into the cell. It's a positively charged ion. And with that positive charge, right, the membrane potential of this cell is going to go up, and the signal can get sent to the brain via the auditory neurons and the cochlear nerve. Okay, so by determining the frequency at which these channels are opening and closing, the frequency at which action potentials are being generated, and where on the organ of Corti these action potentials are going to be generated, our brain can interpret what is the overall volume and the pitch of the different sounds that we hear, right? But it's these hair cells that are responsible for generating the action potential and then sending that signal to the brain. And those are found on that organ of Corti, or organ of Corti.